Embryological basis of neural tube defects. So in this we discuss the competency, that is various types of open neural tube defects and embryological basis. Neurulation is a process of forming neural tube which will become the brain and spinal cord. In humans it begins in the 3rd week after fertilization. The phenomenon is complex, involves numerous cell processes and is often disrupted, resulting in neural tube defects such as anencephaly and spina bifida. Because of the complexity, most defects are considered multifactorial in origin. Primitive state. At the end of the 2nd week, the embryo is bilaminar disc consisting of epiblast cells on the top layer and hypoblast in the bottom layer. Soon the primitive state appears in the caudal third of the disc, signaling the initiation of gastrulation. The process of forming a trilaminar disc containing 3 germ layers, that is ectoderm, mesoderm and endoderm. At the cranial end of the streak lies the primitive node, a depression containing cells important for organizing the embryonic axis. Gastrulation. During gastrulation, epiblast cells migrate through the streak and node, detach and form 2 new layers, ventral to the remaining epiblast. First cells through the streak displace the original hypoblast to form the endoderm, while the cells migrating slightly later create a new middle layer, the mesoderm. Cells remaining in the epiblast that do not migrate through the streak or node constitute the ectoderm. Neural plate. Cells passing through the node to migrate cranially in the midline form the precaudal plate and the notochord. These structures initiate the process of neurulation by inducing formation of neural plate from overlying ectoderm cells. The remainder of the ectoderm surrounding the neural plate forms the epidermis. Index of neural plate is due to inhibition of the epidermis formation by signals emanating from the primitive node. The signal itself involves suppression of bone morphogenic protein and WNT signaling pathways. Convergent extension. Once the neural plate is formed, it undergoes elongation by convergent extension, a process in which laterally placed cells move toward and are intercalated in the midline, that is convergent extension. Recent evidence suggests that disheveled, that is DSH, is required for the process of convergent extension. Misregulation of this gene causes neural tube defects in animal models. Neural folding. Once the neural plate is induced, its lateral borders elevate into folds, neural folds, and these folds move towards the midline to fuse. After elevation has occurred, bending of the neural folds proceed in 2 steps, that is furrowing and folding. Furrowing requires formation of 3 hinge points in the neural tissue, that is a median hinge point, MHP, overlaying the precaudal plate and notochord and extending the entire length of the neural tube. Then paired lateral hinge points, LHPs, along the sides of the folds of the cranial region only. The molecular signaling for inducing median H point is sonic hedgehog. Shaping the hinge points and neural folds require microfilaments, microtubules, changes in the mitotic rates that together create apical constriction and basal expansion of the neural cells. Disruption of the integrity in animal models resulted in neural tube defects. Another factor affecting the cell shape is mitosis. Neuroepithelial cells during neurulation are rapidly dividing and the cell with cells cycle times of 4 to 6 hours. In the spinal cord, many of these same events promote closure of the neural force in the prospective spinal cord regions. However, no LHPs appear. Neural folding. Closure itself first occurs near the junction of the hindbrain and spinal cord at the level of fifth somoid. It then proceeds in zip-like fashion cranially and caudally. After initiation of closure, open regions of the neural tubes are called as anterior and posterior neuropores. Caudally, the zipping process continues until the posterior neuropore is closed. But in the cranial region, a second site of closure appears in the forebrain. Zippering then proceeds bidirectionally from this point to meet the advancing zipper process approaching from the hindbrain as well as cranially to close the rostral part of the forebrain. Cranial neuropore is closed on 25th day while the caudal neuropore completes closure on day 28. The primary and secondary neurulation. Primary neurulation is responsible for establishing brain spinal cord regions down to the lowest sacral levels, that is probably S4 and S5. From this level, caudal word, secondary neurulation forms in the remainder of the cord. In this phenomena, the neural tube forms from mesoderm cells that coalesce and then epithelize. Epithelial cells reorganize around the lumen, forming the caudalmost regions of the neural tube that then becomes continuous with the remainder of the neural tube formed by primary neurulation. The junction between the two processes is well below the site of virtually all occurrences of spina bifida, so cannot be considered a potential factor in the origin of vast majority of neural tube defects. Neural tube defects are common. The term neural tube defect is applied to a variety of abnormalities, most of which result from lack of closure of the neural tube. Most severe are open defects in which neural tissue is exposed. These defects may occur cranially, causing anencephaly, which is fatal, or caudally, usually in the lumbosacral region, causing spina bifida cystica. Closure defects result from failure of the neural folds to elevate and fuse. Neural tube defects and the coverings. Other neural tube defects may involve coverings of the brain and cord, such as meningocele and myelocele, or may include neural tissue as in the cases of meningoencephalicele and melingomyelocele. Bony defects overlaying these abnormalities may be caused by lack of signaling between the underlying neural tissue and the overlying mesoderm and ectoderm. Most of the neural tube defects are multifactorial in origin. Misregulation of any of a number of different genes may result in neural tube defect. Dishevel appears to be an important for convergent extension, and its disruption may account for some neural tube defects. Sonic hedgehog is responsible for floor plate induction and MHP formation. But neural tube defects do not occur when this gene is misregulated, even though an MHP fails to form. Other gene candidates include those in the folic acid pathway. So now we start with the folic acid pathway. MTHFR gene for methylene tetrahydrofolate reductase catalyzes reduction of folate as part of methylation pathway appears to play a role in some of the neural tube defects. It is relatively minor one, accounting for at most 15% of the defects. The search for other genes in the folic acid pathway that might contribute to neural tube defects has failed to demonstrate any other associations. Emphasis on folic acid metabolism as a major contributor to the neural tube defects has occurred because unequivocal evidence shows that folic acid supplementation, that is 400 milligrams per day, initiated 2 to 3 months prior to pregnancy, reduces the risk of neural tube defects by 70%. Folic acid function. Folic acid is essential for maintaining basal cell processes, including novosynthesis of nucleotides for DNA synthesis. Its pathway is especially important for embryos during neurulation because it probably represents their only source of nucleotides. Nucleotides are essential for sustaining mitosis. Cells normally have 5 minutes supply of DNA precursors, and if proper ratios are not maintained, mutations increase, DNA repair mechanisms fail, and DNA synthesis decreases. Since mitosis plays a key role in neurulation, it is easy to consider the link between neural defects and this aspect of folate metabolism. It is also possible that compromised DNA synthesis produced by inadequate folate supply may increase embryo sensitivity to teratogens. Studies show that embryos from mice that were almost completely folic acid deficient had development delays but no overt birth defects, although here was an increase in spontaneous abortions. When maternal folate levels are extremely low, surviving embryos were spared, but were they also more vulnerable to second insult. Folic acid provides essential components for general processes such as DNA synthesis and mitosis. It in increased amounts protects against heart, craniofacial, other birth defects. It also affords protection against some genetic and environmental causes of birth defects, suggesting a general effect of protection to the embryo. Such an effect on DNA synthesis might allow embryos to recover more quickly from insult by producing more cells and by avoiding genetic mutations by providing large nucleotide pools to the DNA repair. Thank you.